So I would love to welcome everyone back. Our next session is a panel highlighting the journeys of Catalyze Awardees. These innovators, will share their progress, some of their completed milestones, and lessons learned in advancing technologies from concept towards commercialization. Our moderator for this session is Dr. Rahul Thakar. Many of you will know him quite well as he is a program officer in the Division of Cardiovascular Sciences at NHLBI. Dr. Thakar plays a central role in the Catalyze program and brings deep experience in translational science and program management, and is committed to fostering the growth of investigator teams as they navigate critical milestones. So Rahul is a great moderator for this session, and I'm really delighted to have him lead the discussion.
Hi, Marissa. Are you ready?
Yeah, I sure am.
Excellent. This is wonderful. Welcome back. I hope everyone had a good break. This next panel is a panel designed to bring Catalyze awardees together to share their stories, their successes, and give you a little bit of insight into some of the, the journeys that they've taken. We have 4 esteemed awardees who will be on the panel. The first is Dr. Zina Kwan. She's the co-founder and CEO of Paramis. It's a startup out of Stanford, based out of Stanford, and research from Stanford regarding electronic skin. For over 45 years, she's led interdisciplinary teams that have transformed advanced materials research into innovative commercial products. She has degrees from MIT and Princeton, go Tigers, and they're all within the field of chemical engineering. Our next guest is Dr. Jaipal Singh, and he's the founder and chief scientific officer of Vasculonics. In previous positions, Dr. Singh has served as a senior research professor and the director of Indiana Center for Biomedical Innovation at Indiana University School of Medicine. Dr. Singh has extensive experience in the pharmaceutical research industry, including 20 years at Eli Lilly, where he led multiple drug discovery programs. And he has expertise within the cardiovascular disease as well as vascular complications of diabetes. He has a PhD from the University of Washington, and he completed his research fellowships at Harvard. Our next guest is Dr. Lucas Ting, and he's the director of R&D at Stasis Medical. And that company is commercializing a rapid bleeding and platelet function test for clinical use and research purposes. Excuse me. Stasis started operating in 2017 as a spinout out of the University of Washington, and its primary technology came out of Lucas's dissertation. That's awesome. Kudos to you. And finally, last but not least, is Dr. Young Sup Yoon. He's a professor of medicine and biomedical engineering and the director of stem cell biology at Emory University, and he's the CEO founder of Caris Bio. He is well recognized for his work in cardiovascular regeneration, where his laboratory develops strategies using stem cells and tissue engineering to repair and restore heart and vascular function. So I turn it over to you, my esteemed panelists. I guess we can start. And if I'm not mistaken, Dr. Kwan, I believe you're up first, and we'd love to hear the journeys that each of you have taken. And as we have time at the end, we'll take and field questions from the audience. And just in case we need to instigate some discussion, I have a few things to get us started with. Dr. Kwan?
Thank you very much for giving me this opportunity to share with you what we're trying to do to create a new standard of care for blood pressure measurement. I'd just like to make sure that my slides are showing.
Yes,
I don't
look great. Thank you.
Okay, great. So better blood pressure monitoring can actually improve many, many lives. Some examples that we've been focusing on, 30 million kids and over 110 million adults visit the ER each year. And one of the first things that happens is that they need vital signs taken. One of those is blood pressure. Unfortunately, it's very difficult sometimes to take sufficient blood pressure measurements because of resource constraints. Another example is about half the adults in the US suffer from hypertension, and they could benefit from better home blood pressure management. Again, this is very difficult to attain in real life, and only about 25% of hypertensives have their blood pressure sufficiently managed. There are many other diseases which also require frequent blood pressure measurements, and we believe that there's an opportunity for us to impact almost every medical interaction. Our dream is to become as ubiquitous as pulse ox is today. And so we've developed a product roadmap to address the needs of of many of these people. All of our devices are built on a common hardware and software platform, and it's based on our proprietary capacitive sensor technology. We've taken a very stepwise approach. We're starting with pediatrics, where right now we're one of the few groups focusing in that space. We're also developing devices, similar devices for adults, initially focusing on maternal care and emergency care. And then finally, we're also developing apps and devices and algorithms for home monitoring. So the science behind our devices, the engine of everything we do are these capacitive sensors, which are very soft and flexible. Let's see, I can show you an example here. But we make these arrays of sensors that are very soft, flexible, and can be made by roll-to-roll manufacturing. And these fit very lightly against the skin, so it's differentiated from a lot of other sensor technologies out there. And they measure essentially, they're very sensitive deflection sensors that look at the motion of the skin above a palpable pulse point. And we use that to collect pulse waveform data, which the pulses are very similar to what you would get with an arterial catheter that's inserted into the artery. And we use the shape of these waveforms, have trained algorithms using machine learning to recognize or to be able to extract accurate blood pressure values from the shape of this waveform. The data are streamed to a mobile device. All of our analyses are done at the local device. And then we can get outputs like this, where this is showing mean arterial pressure compared against arterial line data taken simultaneously. You can see there's very good agreement between the two. And one thing to stress is that for at least some patient populations, we do not need external calibration. So this was an uncalibrated model. And so we've created devices that are wireless and easy to use, comfortable to wear, and provide these accurate results comparable to art-line monitoring. Our first device was cleared by the FDA to continuously and non-invasively measure the blood pressure of critically ill infants. And as I mentioned, it's the only such product so far cleared for the young children. The evidence that we provided to the FDA for this was a 5-site validation study. And we tried, I believe the earlier speaker on machine learning or artificial intelligence alluded to how you need to have a very broad population, a broad set of data to be able to be generalizable. And so we attempted to do that with the different sites. And you can see the types of results we have here with Land-Altman plots. We have commercially launched the product. It's been adopted by 4 of the top-ranked NICUs for clinical use. Whenever we do pilot evaluations as part of our commercial process, we survey all the nurses that have used the device. So far, they have overwhelmingly found it easy to use and have recommended its use. And so they truly believe this could be improved patient care for them. We're a startup, so we care about the revenue stream. We feel that there's going to be a nice steady revenue stream. It's not unicorn scale if we look at just pediatric indications, but it's very nice growth through both wider adoption and extending the indications, both to different types of patients and also to lower acuity care. The numbers become a lot more interesting once you add in the adult products, which we believe we can do with minimal time, cost, and risk, because we build on the same platform that we've developed the pediatric products. The focus of this project, the Catalyze program, it will be our Vocemi product for maternal care, which is our first outpatient product. These are single-use devices that would be used up to 6 weeks, updating once an hour. unlike the baby product, which updates once a second and only lasts for 3 days. We're developing products for prenatal, perinatal, and postpartum moms. And the reason we care is because hypertensive disorders of pregnancy affect about 10% of the population or pregnancies. And they're strong, it's strongly associated to severe adverse events, including one of the highest causes of pregnancy-related deaths. We provide comfortable, convenient monitoring, both at home and also in the hospital. More timely, cost-effective treatment, improved workflow at lower cost. And then the advantage will be reduced risk of early delivery or readmissions. And that's part of the value proposition that we've been building, is to reduce the lengths of hospital stays and reduce the risk of adverse events. And one of the things that we feel is an important value proposition is our devices are very easy to use and comfortable to wear, so people are willing to use it. And that's the biggest problem with a lot of today's current solutions, which is to ask patients to take cuff measurements on a regular basis. Our early results were very promising. This our initial proof of concept. The model was developed for women of childbearing years. We had several pregnant women in our orbit who took data, and we scaled reasonably well. We were part of the NIH RADx program for postpartum care. And so we started collecting data through there. We used that as an opportunity to develop a beta version of our band and Bosimi band, and also more important is to build an app around home use instead of using it in hospital use. With this project, we have spent a lot of time trying to collect the data that we need to develop models for this particular patient population. We've collected data from over 100-some women to date. Most of it has been against cuffs. We actually prefer using art line data because we get a lot more ground truth values and we get to see a lot more longitudinal effects. But most people don't put art lines into pregnant women. We did recently start a study with Stanford where we're trying to collect data from women of childbearing age as well using A-lines. So we've also spent a lot of time developing new models, new algorithms, and this is a general population algorithm for adults of all ages. And we started with an unanchored model, and I'm showing in green the specs that we meet, either the FDA guidelines for accuracy or we meet our own internal specs. And so the unanchored model, we're reasonably good in terms of the mean average error, but we have a lot of variance in the data. We don't have a lot of data to work with to date. But we started working as well as a fallback position of using an anchored model. And so this is where our devices are prescribed by the doctor. And so it's the opportunity to get a cuff measurement in the office or in the hospital, and then the patient gets sent home with our device. And so, and here you can see now that we're meeting the FDA guidelines for accuracy. We don't yet meet our internal guidelines for trend analyses, but that's something that we believe will improve as we get more clinical data.
So your dad has the advantage, okay, which lower the speed.
I'm sorry. And on the right, I show, again, some bland Altman plots just to show the accuracy of our current results at the population level. But you really care about what happens at a personal level or a single person level. And here's a maternal care example. This woman actually was very kind and started taking data in her 17th week of gestation, and then took the data all the way through. She was diagnosed with preeclampsia at 34 weeks. She delivered at 35 weeks. And then finally, we have some data in the postpartum phase as well. The top figure on the right shows our results using our unanchored model. We actually had surprisingly good agreement for the unanchored model for systolic blood pressure. We are completely out to lunch in terms of diastolic. Whereas with an anchored model shown on the bottom, we actually have sort of balanced out the error between the systolic and diastolic values. In both cases, we actually have pretty good, we have very good correlations and slopes for our systolic values. We still need to work on diastolic to improve those. One of my asks for this particular particular audience is there a source where we can get access to historical art-line data for pregnant and postpartum and perinatal women? Because that would really accelerate our progress in getting and improving these algorithms, particularly in terms of trends and correlations. Two areas that we think might be possible. One is for placenta accreta patients and the other is for surgical patients. So if anybody knows any of these sources, we'd love to chat. So a couple words about the company. We spun out of Stanford with an exclusive license to our base technology from Shannon Bao's research in electronic skin. We launched our first product last year. It only took us, since 2016 working on the technology, but we've eventually gotten there. We've collected lots of data from lots of different patient populations, and we think that allows us to envision many different types of exit and licensing opportunities. We have a very experienced team with over an embarrassing number of years of bringing products to market, and would love to work with you. We believe this is an opportunity to create a new way of measuring blood pressure that's good enough for, it's general enough to use with babies, but good enough and comfortable for everyone else to use. Thank you.
Next, I believe, is Dr. Singh.
Okay, let me just get my slides up, and I hope you can see them. Great. Well, thank you for the invitation for us to share our experience, and particularly what I will do is we are a much earlier stage company than Dr. Kwan's company, and I thought it will be helpful to other investigators or the new applicants that what kind of experience we had with Catalyze. And I think that's what I'm going to focus on. So the project that we applied the grant for is a preclinical development of a disease-modifying therapy for pulmonary arterial hypertension. Now, PAH is a really deadly disease, and it is a rare disease, but it is highly prevalent in women. About 70% of the disease is related to females. So, and in terms of market, it's a fairly large market. There are a number of compounds which have been developed over the years. So what I would like to do is first, really kudos to NHLBI to recognize that PAH is really a bad disease that needs development of new therapies. So we got funded through Catalyze. In fact, I was pleased that we got funded first round. We didn't have to revise or anything. It was first round without any revision. We got funded, and then we were also now, with the help of Catalyze grant, enough data to get a SBIR phase 2 grant. So clearly that Catalyze was very important for us to move forward to next phase of the program. And I wanted to share some of the technical and logistic support that RTI was able to provide, because I think this is quite important. So what I will do is try to tell you what we accomplished in a very high level goals, and then also want to share with you how did we accomplish that, and that is with the help of our team as well as the Catalyze collaboration. So in terms of accomplish, we started very early, and I want to show you that we went through lead optimization, and we are now at a clinical stage or clinical candidate stage program. I also want to share that the identification of molecular target that we were working on had been really important and has expanded the scope of therapeutic areas for the company. And as I said, I want to share how we really were able to accomplish these goals through our own team and Catalyze team. So what was accomplished? As I said, PAH is a deadly disease. It's a progressive disease in the lung when the arteries, mostly medium and small size arteries, progressively narrow, and then ultimately really some of the arteries are closed. And that leads to a tremendous pressure on the heart. And ultimately, these patients die of right heart failure. There have been a number of therapies over the years, which are all vasodilators. They dilate the arteries a little bit and give you relief to the patients. But really, these compounds do not affect the disease progression significantly. So there is clearly need to find agents that will really have a different kind of mechanism of action and different response in these patients. So their quality of life as well as mortality can be seriously impacted. So we started out with a simple hypothesis that we had an early hit molecules. And with the award of the Catalyze grant, we were able to go through lead identification that required significant chemistry activity and lead optimization, which was really the major activity. And as I said, we were able to also figure out how our compounds are working. And we were finally able to select a clinical candidate. So this was very important achievement through the Catalyze grant. And as a result, we were able to secure the SBIR phase 2 grant. And now we're going through safety studies on these compounds, and they look really interesting and good so far. So our goal is to begin to think about filing IND in next year and a half. So all that looks very good from the previous slide, but there were a lot of challenges along the way. As you know, drug development is not an easy task. A lot of failures, a lot of issues along the way. And I want to share how the Catalyze team and our team was able to really resolve some of the challenges, and ultimately we were able to succeed in this program. So our first challenge was that the first lead compound failed in the efficacy studies. And that's why I have this, this slide on a blue background, because I was pretty blue at that time. But then we had a lot of discussions with the Catalyze team and our own team about the animal models, the dose that we used, whether the compound is getting into the lung or not, and a host of things. And I actually have an actual slide from that discussion, from that presentation of a quarterly report at Catalyze, that really shows how it worked out. That even though we completed this study, but the goal was not accomplished in terms of getting the compound. So to make a long story short, we were able to figure out how our compounds work. We devised a new screen, and we found new compounds to move forward. So here's kind of hypothesis we started out in the grant, that ADMA, asymmetric dimethylarginine, is a vascular toxin. And the concept was that this toxin is increased in PAH patients. That leads to multiple effects on the vasculature and mitochondrial function. So we were screening compounds based on increasing the DDH, which is an enzyme that degrades ADMA. So if you increase DDH, we'll find that ADMA is reduced, and therefore the progression mediated by this pathway will be reduced. But we were using a phenotypic screen to identify compounds, and we did not really know what the mechanism was. So after our first compound failed, we went back and began to think about actually understanding what the mechanism was. And thanks to Dr. Yang Li, who probably is listening, we were able to identify that the target for our compound was this nuclear receptor called aryl hydrocarbon. And that allowed us to really more selectively select compounds based on this, this receptor. So we devised a new screen and came up with VN1032 as our molecule to test in vivo. And that was quite effective. So we were able to successfully demonstrate efficacy with this new molecule. So challenge doesn't mean you need to worry too much about it, but you have a good team, you can probably solve some of those challenges. So the other very important thing happened in response to our finding that AHR is the target, that we are now pursuing that for PAH, but it turns out that receptor is also involved in obesity, for example, adipose differentiation and energy metabolism. It is involved in pancreatic cancer as well as cognition issues. So now it's a bigger opportunity for us to pursue down the road other indications based on the finding that AHR is the molecular target. So we do have compounds. I think it would be much easier for us to think about other potential applications of those compounds. So this was very important discovery for us. Now let me talk a little bit about how we accomplished these goals. The team composition was very important. So we had a core team of myself and chemist and Dr. Lee, and then we had really a number of important advisors. And given that we are a startup funded by NIH, we didn't want to have employees or experts who are highly expensive. So we were fortunate to have a number of Lilly folks, our friends, were able to join us as advisors and guide us through the developmental process, and they're still on our team. We didn't pay them a lot, thanks for their help anyway. However, I think that was really not enough because the startup needs many other tools. So I wanted to give some examples of how the NHLBI and RTI was really able to help us move this program forward. So here are some examples of issues that were arising and how we were able to solve that. So we started out synthesizing new compounds based on the AHR as a target, but we did not know what the chemical space is. And a startup company does not have all the databases that are required to do patent search and analyze. And so Scott Runyon from RTI was very helpful, gave us all the information, and we were able to move forward with synthesizing novel compounds because we don't want to work on already published compounds. We want to make sure that these compounds can be patented. When our first study failed, we thought the compound was not getting in the lung. So we felt maybe inhaled will be another strategy. So we knew little about inhalation drug delivery. So RTI was able to bring, I would think, the world's expert, Anthony Hickey, to have a discussion with us and really opened our eyes that the inhalation process development is very complex. And it's at least 3 to 4-fold more expensive to develop a product. We also have to have a relationship with a device company. So we figured out that it is important for us to pursue an oral compound. Very important decision to move forward. We also got a lot of help on price and reimbursement from NHLBI expert, Renee Arnold, and a lot of interesting information for investigators, especially from academics, I would say, on commercial strategy. We had meetings with Dr. Flame. It was very helpful connecting to some investors, reviewing our slides, and giving really good guidance for us how to discuss our presentations with the investors. And there are a lot of webinars that Catalyze organizes. They're highly, highly useful. And we learned about how to pitch to the investors through the meetings. So I also want to really tell you it's very important that Catalyze quarterly meetings, they kept us organized and focused. As a scientist, we have a kind of habit of putting the data in a folder and then think about all of the data when we decide to publish it. But in this case, in these quarterly meetings, they really had a very good questionnaire to say, what was the goal? What was accomplished? What were the issues? And more importantly, they always come back and say, how can we help? So this was just fantastic. And I really want to thank all the people who were involved from NHLBI as well as the RTI. When we started out, we thought this RTI group will be a sort of a monitoring group. They will bug us all the time to provide all the information, but it turned out they really are supporting group. They're not monitoring group. So I wanted all the investigators to know that our experience with RTI has been that they are really a supporting group, not monitoring group. So with that, I want to tell you that the Catalyze grant funding, the money is very important. We couldn't do without the funds, but the Catalyze team support was equally important for the success of us going from very early stage to now moving toward IND filing. So thank you.
Thank you, Dr. Singh. That was remarkable. If you'll note in the chat section, we had a nice exchange between Dr. Kwan and Dr. Bakhtar. Feel free to continue e-chats there. Keep the conversations flowing. Anytime we have it, the remaining we'll devote it to questions to the panel. Now, Dr. Ting, the floor is yours.
Great, thank you. So we're Stasis Medical. We're a spin out from the University of Washington. And this technology is really focused on a key problem, which is in most hospitals, you don't have a quick way of diagnosing sort of platelet dysfunction. And the decision to transfuse is always a highly critical one. You have to do it very quickly. There's a lot of positive and negative consequences for your decisions. And you basically only have like a 5-minute window to make this. So a lot of times what will happen is you'll have a doctor, let's say an ER patient comes in, you have to, you see an injury, it might be blunt, it might be internal, they might be actively bleeding out of their body, they might not be. And kind of what occurs right now is doctors will order a test, they'll fire it off to the lab in the hospital, and then they'll just make their decision before waiting for the test to come back. So when we applied for this grant, this was like really a key issue and something that we really focused on within the course of the Catalyze program. So a big thing that this Catalyze group and the whole program helped us do was really fund a lot of technical development as well as run a clinical study, which I'll get into. So at its core, platelets are really, really expensive and they're very difficult to obtain and store in hospitals. It takes 12 blood donors to get one platelet bag, or it takes one person about 2 hours to do apheresis to get that platelet bag, and they're quite costly. Another thing is they only last for 5 days. So once you get the platelets in the bag, they start settling. They'll have surface contact activation. They might activate just touching other platelets. So you really can't store them outside or in a fridge for more than a week. And it's also dangerous to transfuse them. So when you make that platelet transfusion decision, you have to be quite sure. So our product is really focused on this microfluidic platform. It's based on these little microsensors that you can see on the right. There's a little micro channel, and inside the channel there are sensors are sticking out at you in the screen. So those are on the orders of tens of microns, and we flow blood in this picture from bottom to the top. So that blood flies over these sensors. You introduce a lot of shear force. The platelets aggregate out of whole blood, and they activate due to this mechanical response called shear activation. And that causes the platelets to collect on the sensors and then actually contract in. And we measure that physical contraction force by looking at it through a fluorescent camera system. So this is mounted on a microfluidic card that sits inside that box that you just saw. And besides trauma, we're also trying to target a lot of other things like oncology, blood cancers, trying to do optimization of people that are taking daily aspirin or daily antiplatelets or anticoagulants. But trauma is really our beachhead. All right, so like I said, Catalyze really helped us develop the physical box that everything is being monitored by. And this is essentially an automatic fluorescent microscope. It is designed to just contain the card, heat it up to body temperature, do a lot of auto focusing and auto tracking of the sensors, do all the machine vision, report back all the data to the user in a usable way. And then we also do some back-end stuff with the data, which I'll get into in a bit, but that was also Catalyze aim that we were really happy to be funded for. So I did a lot of physical, development of mechanical electrical systems, Like I said, this is a fluorescent microscope, so it's if you're familiar with fluorescent microscopy, it's really sensitive to focusing and motion. You have to make sure that your image stays clean and balanced and planar to your optics at all times. So we spent a lot of the first year within Catalyze developing that out and really honing for sale as a commercial use device. And we're not quite as far as having FDA clearance, but that's something we're intending on going towards the next year. So what's out there right now? There's a lot of devices that do platelet or blood monitoring, like PTINR, two different types of tags, which are viscoelastometers. There's ROTEM, which is also a viscoelastometer. And then the last two rows are kind of aggregometry, what you would consider more like platelet count or clot size counting. And the issue with all these kind of out there right now is they're just too slow. So they take 10 minutes, not counting the time it takes to pipette in reagents, not counting the time it takes to get the reagents from the fridge to room temperature. So it adds up being like 20 to 60 minutes for some of these tests, and that's really not optimal. So we're lucky in that we sort of stumbled upon our technology and figured out we could do this at room temperature with no reagents in just a minute. So stroke of luck there. So one of our big aims within Catalyze was focused on funding a larger observational clinical study because we really needed to gather sufficient data to really develop our machine vision and sort of, I don't want to say staple on machine learning, but it was such an easy thing to take advantage of because we're ingesting all this massive image data that we really wanted to leverage it. So we ran a 750-person study at Vanderbilt University Medical Center inside the ER. It was just anyone coming in the past 2 years to basically anywhere like within 200 miles of Nashville probably was enrolled in our study if they were sufficiently injured. We took their sample of the blood, ran it over the machine, took all of the force and aggregation data, which is what our machine outputs, and we ran it, did all the sort of data extraction, did a lot of fancy image extraction, and ultimately ended up with a model that sort of tells you, within the first 60 to 90 seconds, how likely it is that this person will need a transfusion. Does so with about 80 to 90% accuracy based on our data set. And we're hoping to expand that with a future study and really go into specific disease states within the trauma cohort. So Catalyze, helped us look for resources to develop this, helped us obviously fund all of the software development, which is not in my wheelhouse at all, and really enabled a lot of this technical development. And then after, while this was going on and sort of after, we were still focused on, scale up. Obviously, we're a startup. You have to manufacture this and it has to be commercially viable for this to actually hit the market. So it was instrumental in helping us look for the regulatory resources, figuring out what documentation we would need, getting all the correct ISO documents identified to pull into our own workflows, helped us understand what we had to freeze in terms of manufacturing, what was acceptable within the FDA guidelines. So a lot of advisory, very valuable advisory questions and answers were something that they provided a lot of help with. And they also had a lot of background experience, like all the members of Catalyze seem to have connections or know someone personally that had this exact same past life or past career sort of work. So they were able to connect us and we got a lot of interesting ideas that way as well. Right, so I mentioned the software development. And yet another thing Catalyze helped us with was figuring out understand who our customer was, because we were very siloed and focused on the trauma market because that was the most obvious market for us. But we in our sort of quarterly updates and the annual meetings, we really got some ideas about what other customers might be out there. And so we developed some web tools to help customers kind of see remotely what they would be looking at if they were using this tool without actually having to ship them a box and set it up for them. So this is something that I think is very valuable, and definitely I would recommend to anyone is use Catalyze for their customer connections. So this is just a sort of web demo that people can look at to sort of simulate tests for their own patient set, but it provides them actual data from our actual tests. But it was a great way to get feedback from people that might be interested but wasn't sure what the machine does because it's kind of a novel output. Right, and then the commercialization support I alluded to, but the pitch competitions, the annual meetings, having to create a 60-second pitch or a 5-minute pitch is very valuable because like the previous two presenters also alluded to, as scientists, you're not used to doing that, and startup business talk is very different than scientific poster or presentation talk. So having to focus in on that, I guess, update your vocabulary, be aware of what the terminology and what investors want to see is all very important and critical as you move forward. And it helped us, raise about 1.3 million in a Series A, and that was, I think, really only enabled by Catalyze because you just don't have these opportunities very often, and so having a pitch sort of be honed in through the Catalyze program and have that ready to go, and then practice it aggressively for like dozens and dozens of investors is going to be such a important part of your own startup. So we're currently partnered with a commercial distributor that distributes other blood testing device. We have our own sort of marketing and sales effort, just from people that we know local to Seattle. But we've also partnered with academic labs, so both with Vanderbilt as well as a bunch of other labs around the country, both trauma. We're looking to go into dialysis and oncology. And then we still use the Catalyze resources to understand the sort of international markets, because that is something that we also want to expand into, is the EU, and get all the important EU waivers and classifications, because that's also a large model or market besides the US. And the regulatory landscape is very different. So Catalyze, again, has the resources that really helped us understand what the difference is between the EU and US regulatory landscape is. And it seems like there's quite a bit of shifts in the past 2 years, which we wouldn't have known about at all without some expertise. So last thing I want to say is, like, all the grant officers and program officers were extremely supportive the entire time. And as a startup, you're usually like just either by yourself or with a small team, and you really don't have cheerleaders. So I think it's so valuable to have like a constant source of support that is just kind of always going to encourage you no matter what. So anyway, thanks.
Thank you, Dr. Ting. That was great. And our final snapshot vignette story, I guess, comes from Dr. Yoon. So Dr. Yoon, let me hand the podium to you.
Hello, my name is Young-Soo Byun. We have been developing human iPS-derived endothelial cells as vascular therapeutics. I'm going to go over how Catalyze gave us help in developing this product. It's now at the clinical phase in Korea and the preclinical phase in the US. So we're going to go through very quickly the journey that we have had with this Catalyze team. So this project aims to develop cell-based therapy for treating patients with cardiovascular diseases using patented iPSC differentiation technology. Human iPS-derived endothelial cell is the final product, which is to grow new blood vessels called neovascularization. So we are targeting peripheral artery disease and myocardial ischemia and others. But our first target disease is PAD because it's a low-hanging fruit relatively. So when even the complication happens, it's rather safer. So as many of you know, ischemic cardiovascular disease are the most common diseases. The number one is ischemic heart disease, including myocardial infarction and peripheral artery disease. Both started with atherosclerosis, meaning it blocks the arteries, and then once it's completely blocking, then the tissue damages happen. In case of peripheral artery disease, multiple arteries are affected, and then symptoms and signs happened. So there are a lot of options for treatment. For example, interventions in case of heart, the bypass surgery, and in the worst case, we have to go to heart transplantation. In case of PAD, when the disease is really severe, all other options are used, then the patients need an amputation. So around 10% of those patients are still untreatable with the current options. So the idea for us is that for both diseases, when the vessels are blocked, we want to grow the vessels bypassing this blocked area, which is called vascular regeneration or neovascularization. To do this, we need the fundamental cells, endothelial cells, for growing the blood vessels around this area. We have developed the differentiation technology into endothelial cells from human iPS cells, which is published in Circulation 2017. This is a clinically compatible protocol. We started that from the very beginning, the clinically compatible protocol in mind. So the idea is that when the PAD patients come, then we take a 5 to 10 milliliter blood, and then generate iPS at GFE facility, and then differentiate them into endothelial cells and put it back to autologous manner. So the thing is that we're pursuing autologous because we want to avoid the effects. As you know, a lot of cell therapy failed. There are multiple issues. Number one, I think, is an immunological issue, and the other is the choice of the cells. Previous studies showed that adult stem or progenitor cell didn't work well. And the issue here is that we already started a large amount of work in Korea back in 2017. However, the issue is that if we want to move the product in the US, there are different ethnicity, and also the race and the disease patterns may be different. So we have to compare the efficacy with the cells developed in the US. So in this Catalyze program, we propose to develop autologous iPS ECs and its validation. And our 33 phase, we wanted to determine this iPSCs as a lead product. So basically determine the therapeutic effects on animal model. So with the guide of the Catalyze team, we developed this idea. We originally thought that we want to do this, generate the cells in the US. However, Catalyze team has been tremendous in helping us, giving an idea that we can generate the cells in Korea. So the cells can be frozen here and then shipped back, shipped to Korea. And once the cells are generated, and then the cells can be brought back to the US and given to the patients. So this idea was totally an awakening experience for us, because otherwise we had to spend a ton of money for just, renting the space in the US, and then, hiring people for generating the cells. So it's going to take, millions and millions of dollars. And we were able to save this much money through the advice. So this is the data. We generate around 22 iPS cells from different ethnicity and race, and then we compare their pluripotency and also the karyotyping and the vector clearance, and also we differentiate them into iPSCs and compare their identity, function, and also the genomic stability by low-pass whole genome sequencing. We also got an idea of this the whole genome sequencing from during the discussion with the Catalyze team. Initially, we wanted to do only the simpler CGH array and SDR, but later we had found that this more sophisticated genome sequencing is required, and we got help from here as well. And also we determined the therapeutic effects on animal model, which is a hand ischemia model. But here we got help in determining this dose for therapeutic effects. This 3-dose dose escalation study was asked And then we got much help from the Catalyze team for determining the final doses. And with this, the red are the cells that we injected into the hind limb muscle, and the green are the vessels, and we really saw the blood vessels formed, and then they are connecting the host vessels. So I don't think many people have seen such beautiful vessel formation from the injected cells. And then thanks to this studies, we're able to come this far. And in Korea, I'm sorry. Yeah. In Korea, we already filed a IND and then got an approval for first-in-class iPSC-EC therapy for PAD patients. And with the help of the Catalyze program, we already had a successful pre-IND meeting in July 2024. And as you can see, there are several people who joined from RTI team. And even after the pre-IND meeting, we had our remaining, agenda and then Catalyze team gave us help for, guided discussion and other help as well. And we are further pursuing this Catalyze preclinical application for frozen cell study. We applied this preclinical service. In this case, we want to characterize the in vivo characteristics of the frozen iPSCs as well. Thanks to the grant, we were able to establish a lab in Atlanta at Emory University in the space of lab to lunch, and we also have a small office in Boston as well. And we also were able to receive NHS TTR grant about 2 weeks ago. In this journey, this Catalyze team is unarguably the best that I ever got in any grant program. NHLBI program officer here, Thakar, Dr. Thakar, and PX, and also all this RTI team has been tremendous, helpful to guide us. For example, for manufacturing aspect, we didn't know where to generate the cells, and then they specifically guided that the cells can be generated, and then also the scope and guidance of a self-freezing studies. In terms of regulatory meeting, we originally wanted to have an interact meeting, but by the advice of Catalyze team, we skipped this one and we directly went to pre-IND meeting, and there were multiple separate regulatory meetings with the Catalyze team. And even after, RTI staff attended our pre-IND meeting together. So that really, really gave us a ton of help. And also we were able to establish a company in the US, and then, NHLBI program officer and director gave us help in terms of how to apply STTR SBI. And finally, this grant was funded. And also that those meetings and the guidance really helped me to keep our milestones so that we can really expedite our progress in the US. So I really appreciate all this team and this program. This was the best experience myself in grant funding and management. Thank you all.
Dr. Yun, thank you for taking us down that journey and showing us the path you took. In listening to all of these stories, I walk away realizing when I look at it from our program standpoint, Catalyze is very much bespoke for what NHLBI requires and needs. And what the program is able to offer you, mainly through our partners at RTI, we're able to do the same for you to meet your needs. It is something tailored. It is something bespoke for what you require. It's a well, if I were wearing a hat, I'd tip it to y'all. So thank you so much. We have about 5 or 6 minutes where we can probably field some questions from the audience at large. Perhaps if you want to raise your virtual hands or type it in the chat box. I'd be happy to relay that. If not, I have a couple of questions I'd like to run by y'all. While we wait pardon that, I see questions arriving. I'm going to go left to right on my screen. Dr. Prasad, will you please go first?
Yeah, thanks, Rahul. Very nice presentation, Yunsub. Very interesting. So the question I had was that I think I should thank RTI because RTI has been a great support to us, giving us new directions each time we present in our quarterly meetings. But the question that I had was, what were the challenges you faced when you had to actually ship the samples to Korea and get it back? There's something that, as we start making our own compounds, that's something that I'm thinking of as I'm worried about it. What challenges did you face so that we could also navigate the same kind of process?
Yeah, there are various sort of hidden steps behind. For example, what kind of the containers you're going to use, what kind of frozen methods you use, what kind of shipping companies you are going to use, and how you can deliver, and the timing. And also these days we're concerned about the tariffs, hopefully not the customs clearance and all those things. So this has been a multiple step. So you need to really sit down and really draw each step and approach the actually also in the early phase, Catalyze team gave us a help what to do in this process. So you can discuss that as well.
Thank you.
Excellent. Jeremy.
Hey, everyone. Thanks for the great presentations. My question is also for Dr. Yoon. From a regulatory strategy and a CMC strategy, I'd be curious to know your how you're defining your drug product versus your drug substance versus the reagents, particularly the reagents that I imagine you need to use to do that transformation, because there's probably more than one of them. There's probably culture medias. And then what level of GMP you need to follow? Like, you need to do CGMP for everything. Can you do GMP grade for certain steps but not others?
I mean, that each step, I mean, if I want to explain all the steps, I will take another hour. So, so simply, there's a FDA guidance there, definitely. So you take a look. And then also, we had a lot of meetings in ISSCR and then ISCT. So their guidelines can really help. And then I encourage you to join such meetings so that you can get an idea. Most recently, we went to 2 different meetings, and then there are people who are like us, so we can all share this ideas, what we can use, for I mean, it depends upon what stage your company or product is, so that you can in your really pilot product, you don't have to use the CGMP. You can use just a regular lab, but over time you should go to CGMP and GMP.
Excellent. Any other questions from the audience? I have one, just if I could relay to the panel at large, no one in particular, but if someone has a thought, please share. It's not lost on me, or I think anyone who listened to you, that it takes quite a team in order to bring your work forward. What are some of the considerations that you take in terms of building your teams out to have the right types of people, but as well as just the right people to work with?
Well, I can take some of it. For us, I think availability of highly experienced people in town was very, very useful, because some of these folks have worked at Lilly for many years and they know how to develop drugs. So consulting with them was quite valuable. Given the funding, we kept the team lean and mean. That means we wanted to spend all the money we can on research than hiring consultants and advisors. And so that also helped us quite a bit. I think CEO is very important. If you don't have CEO, commercialization becomes very challenging.
Indeed. I think with that, it's my opportunity to hand this back to Marissa, and we should probably collectively thank our panelists. Thank you so much. Thank you so much for taking the time to prepare this and share this. And I believe we have another panel coming forward. Marissa?